Okay, so today we're going to start talking about visualization. So visualization is sort of critical to, I think, any process of data science, and try and give you a sense of why, based on what we talked about before, the sort of locked-in nature of our intelligence. So the best estimate, or maybe it's not the best estimate I could find, but an estimate I found that was plausible and reliable, is that the optic nerve, which I'm not calling a nerve here because it's not really a nerve, it's an extension of brain tissue that is processing information as it takes information into the brain. And that has a communication rate of around 8.7 million bits per second. So that's like the first sort of wave of Ethernet. It's about, I guess, 1,000 times slower than modern Ethernet and much, much slower than you get in your camera. But between the sort of CCD sensor and the processor, but it's a really fast compared to our ability to communicate orally. So our visual system is extremely impressive in terms of its ability to absorb. Of course, we can't send back information through light, electromagnetic radiation. That would make us beings with Wi-Fi. But we can absorb information in that way. And that presumably has had a massive effect on the way our intelligence absorbs things and processes things, including the fact that we can both be looking at the same image and we can talk about things in the image, for example, discuss what they are and describe them. And then if I mention that object to someone else, they've been able to absorb that object at high bandwidth. So they have a very rich picture of what I'm talking about if we talk about, for example, a cat or whatever we might want to talk about. Now, despite the very much larger bandwidth of our optic tract, the eyes still aren't high enough bandwidth to absorb the entire visual scene in front of them. So what they actually do is saccade. So you can sort of tell that if you put your hand there, like I can't see my hand properly at the moment, but I have an impression of seeing my hand because at any moment I can go and grab it with my eyes. So there's this very interesting point that this is not a passive reception. The way you're viewing the visual scene is through scanning. So your eyes do what's called saccading. And you can even show, there's experiments that show, for example, if you give someone something to read and you track their eyes, and so the only place on the book that you actually put the real words is right where their eyes are focusing. The rest of the book you jumble. People don't notice, right? Because as long as the piece in front of their eyes, which is where all the sort of detailed cones are for receiving the image, is in focus, the assumption is everywhere else is in focus. And as long as the eye can saccade and move and check, then you have this impression that everything is constant outside that regime. So someone who looks at this a lot is a gentleman called Donald Mackay, who I never got to meet, but I did have an extraordinary thing. I think it was in this lecture room or the next door lecture room where we were celebrating the life of his son, David Mackay, who unfortunately died some years ago, but he had cancer. So we had a celebration of his life. And his mum was there, Valerie Mackay, and she actually edited this book. And I ended up speaking a lot to Valerie about Donald. Donald was, he was graduated sort of early in the war and started working on radar during the war, but after the war, he ended up working in this group known as the Ratio Club, which was people like Alan Turing, Donald Mackay, a bunch of others. And he became very interested across his career in the eye and this notion of the eye as an entity that is very active, that it's grabbing at the world. So this is his book. And he actually sort of has this whole sense of that's also what your brain is doing in some, which I quite like this idea. I'm not sure how valid it is in the psychological literature, but just as your eye saccades, your attention can saccade, in some sense. I kind of like that idea, although that's just me speculating rather than some luminary in the field. But as a result of this, sort of visualization is very intimately connected with human perception. But of course, it could also be vulnerable to manipulation as well. So just as when we talked about sort of conversation between people, when we think about social media, in fact, a lot of the ways that social media does manipulate you is through your visual system. So that sort of the little message tag, how many unread messages you have, is always flipping red, isn't it? Because it's trying to attract your visual attention. And that turns out to be for a special reason, because your perception of red, and there's this amazing book which starts talking about your senses by Kevin O'Regan, who was inspired to work in this area by hearing Donald Mackay when he was at school, talks about your visual perception and what your feel for the world is and how it relates to the physics of both your receivers and the incoming, whatever you're sensing itself. And the point that Kevin O'Regan makes about red, I'm going to try and get this right, let's try and use the visualizer for this, is that it has a particular property. And I'm not sure I'm going to capture this well. What can I log on for? Let's go for that one, actually. But so you've got your eye, and it's perceiving incoming photons which have certain energies. Now, within your eye, there's low frequency. Well, these photons have energies going across the frequency spectrum, the frequency of electromagnetic radiation, and you didn't tell me I had to zoom out. So you've got sort of what happens in your eye is your cones are sensitive in different ways. So you have red, green, and blue. So incoming, this is some rough representation of how they respond. So incoming photons that hit here trigger both the red and the green. And you end up perceiving something between them, you end up perceiving yellow. But also, if you simultaneously get a photon from here and here that's mixed, you end up with the same effect, you perceive yellow. So you can either perceive these colors because of their pure spectrum or their mixed spectrum. But what's interesting, well, there's a couple of interesting things. That first of all, that there's a response from the blue in the red regime. So that's why you perceive a color circle. That's why you perceive that the colors go around, because actually as light starts becoming perceivable, it triggers the blue to some small extent, which is why you don't have this perception that there's a sort of discrete change between blue and red. But what's interesting about this is you are visualizing what is, I mean, it's a multi-dimensional signal, but in terms of color, it's one-dimensional, and you're visualizing it in 3 ways, right? You know, an alien sort of perceiving color might perceive it totally differently, right? Because we're choosing to place these sensors here. We're not choosing to. We've evolved to place these sensors in some particular interesting way. So this is a form of processing of data. And what's particularly interesting, according to O'Regan, is actually when you look at the whole physics of this, and this is certainly his thing he studied, is when you have the sort of actuality of how these sensors work, you can think of this as a 3x3 matrix, that you've got incoming what should be perceived as red, green, blue, and what you actually perceive as red, green, blue. So you get a 3x3 matrix. This is my rough understanding. I don't have a detailed understanding of this. And what O'Regan says is that for primary colors, this matrix is low rank. And in fact, for red, it's rank 1. And what this means is that red sticks out, because when you see red and you move your head around it, it doesn't respond in the way, or as the shadows fall on it, or whatever's happening, as the light's changing. It doesn't respond in the way you would expect. So this is why you have these primary colors, according to O'Regan, that I think, and it's across the, there's also something about cyan and whatever, that you get the same effect. But red has this particular effect that this matrix, if I've understood it, is rank 1. Now, this is interesting because this is saying whatever's coming in here, in theory, should be 3-dimensional. It's being projected down to 1-dimensional. And kind of, that's what we're sort of talking about also today. Although we use our eyes to visualize things, our eyes are also themselves an instrument of visualization, which have all these properties that taking high-dimensional things, however many photons, and projecting them down into some space that we can understand. So we're going to start by thinking about one way of visualization that is discrete latent variables. So So in a discrete latent variable situation, what we have is we have some object or data, and we have a sort of choice. The notion is that there is some categorization of this object or data. And in a discrete latent variable situation, we think of it as a discrete label. So a discrete label might be, pen or cat or some type of discrete thing. In fact, I'll go back to the projector just to, oh, you don't want to, no. It's to tell a small story, which always reminds me of the difference between these two things. So when my son was about 4, I read something that said that you could teach children about the nature of infinity by telling them that asking them what the biggest number is, and then pointing out there was one above that, and that they would very quickly learn about this concept. Well, this turned out, in my poor son's case, he was subjected to be trying this to be not very true. So he was very resistant in an interesting way to this idea, but he also got annoyed. So I would try it a lot of different ways. So I tried something else. I sort of said, well, how many times can you go around this circle? And he said, once, which I thought was a great answer. And then because I'd drawn, he was quite young, he was probably less than 4. He was quite young because I'd drawn a shape. He started drawing triangles and, I don't know, whatever else he was drawing, rectangles. And so I said, okay, draw all the rectangles. And so he sat there for a while and he was doing this, and he sort of was doing these sort of things, I don't know, like this. And he did it for a while, and he said, right, I've done all the rectangles. And I said, well, how many are there? And he'd drawn 8. I remember quite well. He'd drawn 8. He said 1, 2, 3, 4, 5, 6, 7, 8. And I sort of said, aha, well, what about this rectangle? And he pointed to the nearest one by Euclidean distance and said, that's one of those. This is, I think, conceptually very, very interesting, because I think he was absolutely right. It is one of those. And what he's talking about is the difference between, so rectangles live in some, there's properties to rectangles. They live in a space of drawings that could be very, very high dimensional, but implicitly they live in a low dimensional space. If we remove the rotation and whatever else, we can even remove scaling, but let's keep scaling in. What you get is a sort of a width and a height. And every rectangle lives on this width and height plot in a continuous way. And that was my sneaky way of trying to persuade him that there were uncountably infinite rectangles. But of course, what he did is he covered the space in crosses. And this is a process that we sometimes call vector quantization. And it's actually really what's going on in deep learning at the moment as well. There's a load of this going on for model compression, right? And the idea is, well, you don't need the sort of fine-tunedness. And it does seem that perceptually, certainly my son was very into discrete representations of objects and very resistant to this notion of continuum and infinities. So clustering is kind of when we're trying to do that. So even though we might not agree, or we might mathematically know, we might look at the rectangle and say, well, this is clearly living on a continuous space. It seems that perceptually, we kind of like the idea of reducing it to a discrete space where these crosses might each be labeled. So have you presumably seen k-means clustering before? Yeah. So we'll just have a look briefly at what that looks like, just as a reminder. The problem with it is it's very susceptible to initialization. So actually, if you do use it in practice, have you heard of k-means plus? It's an approach that really improves it, but it's an initialization approach. So actually, initialization gives you a really good clustering. So if you do it in the standard way, which is initialized with random centers, so you take your data, and what I've done in the next thing is, when I demonstrate it, is just select each data point as a center randomly. And then what you do is you assign every point in your data set to the nearest center. So in, I should probably just leave this up. In my son's case, Fred, with his, let's Oh, that's I don't run around. It's very confusing because it says left and right on here. I can't, was it your left, my right, whatever. It should just have the arrows on it. So So what's going on in this case is basically what something like what Fred was doing. So if you have all the rectangles as a data set here, they should cover a continuous space. But if you start creating, if you choose this as the center, and then this is the center, well, you'll get some effect where that and that are seen as one of those, so one cluster there, and another effect where those are all seen as one cluster there. And the way you do that is you simply do an assignment of the points to their nearest cluster, and then you update the centers as the cluster means. So you do the assignment and then update the center. This turns out to provably converge. It also turns out to be a sort of cheap version of a mixture of Gaussians fitting. And typically it has this sort of minimized squared, sum of squared distance between your sort of center and the cluster. So if you're trying to sort of quantize a space, and I do think that it's not a clustering algorithm, K-means, right? Because in some sense, K-means isn't what we might think of, I think of as clustering, although not everyone would agree, is that here's two clusters, right? Whereas in K-means, it's quite happy with the situation where these are not disconnected. So the two clusters here assumes the density between the two clusters decreases, right? And that's what we talk about as star clusters. Whereas in vector quantization, what we think of is, well, the whole space is sort of continuously covered in points somehow. And K-means doesn't actually look for a reduced density at all. It would find one if it was there, but it's not sort of scoring on the basis of that. It's scoring on the basis of how tightly it represents the points. So in some sense, I kind of think of it as a vector quantization rather than clustering. And it's interesting because these ideas have a lot of history in terms of like Platonic ideals from Greek philosophy about what is the ultimate chair, what is the ultimate bird. But actually, the interesting thing is you look at data itself, you find there is no sort of easy average that can summarize. And this is one of the points, the problems, that these prototype points actually don't represent very well unless we choose the space very carefully. It will require multiple prototype points to represent the diversity of a class. So it's often not the case that we find that we've got these Platonic things. So we can just choose our square distance and minimize it. And then our objective function is the minimum square distance between those points. As I say, it's very initialization sensitive. It's very quick and simple to implement, but very, very initialization sensitive. So I'll sort of give you a sort of sense of that in here. So here I've initialized, I've got to switch onto there, so apologies, yeah. So what I've done here is initialize the K-means clustering in the following way. So here's some points. I have sampled them from Gaussians, so they're roughly separated. I've chosen a random example, 3 random examples to give me 3 centers. And then I've run the code. And the first thing it does is allocate all the closest points to that center. So in this case, because there's no center up here, all of these points are going to get allocated here. This one gets these points here. But now what's going to happen next is we update the location of the center to the mean of the points that are associated with it, which is going to cause this one to jump up here, and this one will move in there. So we will get quite a rapid convergence. Okay, after one step, we've moved the means, update the allocations, do the same thing, move the means, update the allocations, and then actually we're getting something that is pretty close after 3, 4 iterations. So I think I can go all the way to 9 iterations. It's not moved. So it's converges very quickly after 4 iterations. But interestingly, I have this code. I can rerun this code every time I create the lectures. And when I reformatted the lectures, I had to extend the number of iterations because the first version, one version just hadn't converged. And you'll find the code in the notebook associated with the lab. You can play with that yourself, try different initializations and see how quickly it converges. Or you can also, I like Alex Iller's, oh, that's a bit old now. I should maybe check, but I still like it. K-means clustering lecture. Alex is at UC Irvine, and I've provided a link to that if you're interested in seeing that. Now, K-means clustering is interesting. I don't think it's that widely used in visualization. It's used for loads of things. It's not the sort of thing that's very useful if you're trying to communicate something about a data set. So if you're trying to communicate something about the data set and you just say, well, these are the clusters and the data's high dimensional, sure, there in these two-dimensional cases, we can see something about the clusters, right? But if the data was two-dimensional, you can visualize it anyway. But what is very interesting is hierarchical clustering. So what you can do instead of clustering things according to just k-means is you can do a different approach, which is super widely used in biology. And I think it's really worth doing with data sets. So if you have a data set like this, where you've got 3 or 4 clusters, something like this, and then something like this, I don't know. What you do in hierarchical clustering is it's a sort of non-parametric approach. And what you do is you greedily go through, so it doesn't give you a guaranteed result every time, selecting a point and then finding its nearest neighbor. And then when you do select such a point, so let's assume you've chosen this one, you merge, so you find the minimum distance between points. You find whichever those points are, you merge them and replace them with a modified point. And you sort of keep doing that, recursively repeating that, until you've got just down to 1 point, which should be at the mean of the data. But the way you then visualize that is you sort of show which points you've merged as a dendrogram. And you show what the distance was between them with the height of the dendrogram. So if you were merging 2 points that were very long way away, you bring them together like that and replace them with a new point in your dendrogram. If you were merging 2 points which were very close, you sort of show them like this and do that. And of course, once you've got this point, it may be merged with this point here, but it may be very close to that resulting point because you can have a situation where you've merged 2 like that. Oh, I can't draw that. But anyway, so the dendrogram goes up until you've got down to sort of 1 point. And what's very cool about that is if you know something about the data, if you're a biologist, you can see patterns in this data. So I've given you a data set to play with in the notebook. In fact, I'll make a shift over there in a moment, where we'll look at the dendrogram of that. And the sort of patterns you might see is things like this, where you have a very tight together set of points here, but then you have a very tight together set of points here. And then they merge like that. And what this shows you is that there is a gap between the clusters because these long bits here show an interesting point of separation, right? What you can do is you can cut here. And biologists love this because they have often an instinct for what they're seeing in their data. So people used to do this enormous amount of gene expression data, right? So with gene expression data, it's 30,000 genes, maybe 50 individuals, right? And they've done some experiment where they perhaps perturbed half of the individuals with one treatment and left the other half of the individuals untreated. So they're looking for something where there might be 2 clusters, right? And they're interested in the cluster labels. So each layer here, as you cut different numbers of lines, has a different set of labels. This is a clustering with 2 clusters. Down here, there's clustering with 1, 2, 3 clusters in it, right? So the biologists themselves can choose where to go and look at that visualization. And you also get a visualization of the data set. So this is the oil data. A little bit off screen at the bottom. How can I get that on screen? Can I zoom out? There we go. So you can see in this oil data, which is 1,000 data points, this pattern I'm talking about. There are actually in this cluster three flows, three different flows. And what it's done is I've labeled the different clusters according to their flows, and it's actually recovered them. If you look at the, so I'll describe what this data set is. It's 12-dimensional data of oil flowing in a pipeline. And the sensors are like density sensors pointing different places, right? So the oil flows in I think, at least three different ways. I think primarily three different ways. One is a laminar flow, where it's like gas, water, oil. One is an annular flow, where I think it's on the outside is water, oil, gas in the middle. And then the third is a turbulent flow. And you're seeing these measurements of these different flows here. And what you're seeing is that the hierarchical clustering algorithm extracts these three clusters with this sort of dendrogram. Sort of down here, they're really close together, right? Only going up a little bit. But as you go up the dendrogram, this cluster is quite a long way from this cluster, right? Does that make sense? It's something that, like, in some sense, machine learning people really dislike this because there's not much theory to do around it, but practitioners love it. You won't see this often talked about in machine learning conferences or even stats conferences, but you'll see this constantly in biological papers. So it's sort of related to ideas of, I mean, trees are interesting because they are actually related to the way we think things came about. Now, the biologists use those trees on transcriptomic data, but they're not believing that there's necessarily a tree behind that. They're just using it as a way of visualizing the clusters, as a way of trying to select the right number of clusters. But of course, there's, there is a dendrogram behind us. So whatever it is, chimp and gorilla and human. If you go back evolutionarily, you find a point where there's a common ancestor here and a common ancestor there. So dendrograms are interesting as really being the concepts behind things. But that doesn't necessarily mean that when we're building a dendrogram from data that that's what's going on. It's just a sort of visualization. And in that way, it can misrepresent. So there's also really interesting work on concepts in the psychological literature that tries to look at how humans generally do, whether they do conceive things in trees or whether they conceive things in other ways. And I think there's various things where people are showing, oh yes, sometimes people do conceive things in trees, or other times perhaps they're not pure trees. There can be links like this in what's going on. I did some work with a guy called Josh Tenenbaum, so my last knowledge of this is from like 12 years ago, so I'm probably very out of date, which was all about these sort of psychological experiments and trying to understand how people are conceiving these different objects. So hierarchies are interesting, but they may not be a hierarchy because it's actually there in the data. It's just a visualization. So for example, we could also envisage using these things for things like e-commerce products. So splitting products into electronics, phones, and smartphones. There's definitely a hierarchy there. But it does have this sort of problem that you can get this sort of thing where money products might belong in multiple clusters. Running shoes are both sporting goods and clothing. So tree structures are often much too rigid for natural concept learning. And concept learning is often more flexible. So there's overlapping categories, and we learn abstract rules and build what we think might be causal sort of theories. So other clustering approaches I'm not going to talk about today include things like spectral clustering, which is very cool. So one challenge with k-means clustering, and also the hierarchical clustering we're doing here, is it doesn't actually account for this sort of property that is often, you'll see sort of people building data sets, artificial data sets of this type, where there's some sort of data in a ring, and then there's data in a center. And the sort of sense, the human perceptual sense is this is all connected. But k-means clustering doesn't look at that gap. That's the thing I said in the beginning, right? It doesn't look at the fact that conceptually, although we can see the Euclidean distance between these two is perhaps less than the Euclidean distance between these two, we perceptually might think of that outer ring as one cluster and the inner ring as another cluster. Again, we start seeing the difference between vector quantization. If we're going to vector quantize this, we probably want points all around the ring. But if we're clustering, we might want this to be separate. So there's a technique called spectral clustering, which is actually allows you to account for this type of structure. And whenever you have these sort of spectral clustering techniques, what's typically going on is underpinning what you look at, there's some kind of graph being built. And the graph connection, and what you're doing is distance along some approximation to a graph distance. So even if we were to just build a graph here, and we were to look at the eigenvectors of the adjacency matrix of the graph, it would already start picking out the two dominant directions. And that's because they are closely related to notions of distance by random walks along these graphs. And we're not going to go into the details of that, but because of that, there's a large amount of work on techniques that use graphs and look at some form of eigenvector structure of the graph and then do dimensionality reduction, or in spectral clustering case, clustering in that. What they actually do once you've got this structure is you do some form of k-means clustering on the new space. Okay. So we're going to switch now and look at continuous latent variables. So continuous latent variables are the things I sort of talked about at the beginning. And like I say, conceptually, it seems we often, or at some level in our brain, we like to discretize things. But in practice, many things are sort of clearly continuous. So the example I like to use for this is thinking about handwritten digits and thinking about manipulations on the digits that we might see as maintaining the nature of the digit. So this is a digit from the USPS data set. And it lives in this very, very high dimensional space. It's like, I think it's like 3,000 pixels in there or something like that. So the number of possible images is 2 to the 3,000 or something like that. Some incredible number of images. So if we were to sample uniformly across this space, so just by sort of sampling each pixel independently, the chances that we would ever see the original 6 are 0, right? So that there's 3 examples. See, none of them with the 6. That's proof of 3. But you know, conceptually, the 6 clearly does not live in the space that we've given it to live in, right? You know, actually, even QR codes don't live in that space because QR codes are restricted parts of that space, but they look a little bit noisier. In fact, I watched an interesting YouTube video on them that said that actually they actually work to make sure they look a bit random. They try and remove patterns from QR codes. So that QR codes don't even live in these spaces. So what does that mean for us? Well, we can have a simple model of a digit where we rotate a prototype. And what I'm going to do is just go to the notes. Let's have a look. So let's go to the notes. Oh, too much force of habit. Lawrence Watt. Oh, have I got that wrong? What have I done? Oh, I've put it twice. So yeah, the lectures and this lecture here, in fact, this is the sort of cover sheet for the lecture. So what we look at in the lecture, in the notes here, and this isn't set, this is just to give you examples of me doing things. And also, by the way, you know, this is me coding to demonstrate what things are going on. Don't just cut and paste my code.
So that's the k-means with the code you use to generate. There's the clustering of the oil flow. And there's the digits. But what we can do is actually think about sensible models of the digits which would retain the nature of the 6. So what I've done here is I've taken that 6 and I've rotated it through 360 times. And I've created a data set of 366s. And then I'm projecting down that data set, which lives in these 3,000 dimensions, onto 2 dimensions and visualizing it for you. And what you see is a circle. And you see a circle. I mean, the first time I did this, by the way, I didn't think, oh, and it's definitely going to be a circle. I mean, in retrospect, it feels, oh, yes, it should be a circle. But you see a circle because the operation you're applying to the digit is a rotation. Now, the reason why things don't sit purely on the circle is because there's some interpolation. I can only rotate at 90 degrees precisely, right? I can't rotate precisely an image at 5 degrees. So there's some sort of bilinear interpolation having to go on to try and recreate the pixels, which is why it's a little bit noisy as well. So it's not a pure circle. It's like a circle with a little bit of noise. And just to sort of highlight, okay, so some of them, let's argue they're 9s. So these are 6s and these are 9s. But the point being that this data lives on these lower dimensional, oh, sorry, I'll put that back up on there. Lives on these lower dimensional manifolds. It doesn't just live in this very high dimensional space. And the manifold hypothesis is the idea that objects like this, and these can be very nonlinear transformations, an object like a 6 consists of one or more prototypes. Think of a 7, for example, having different styles of 7 or a 4 or whatever, which then can go through transformations which don't change the nature of the object. And those transformations could be rotation. They could be other things like thinning of the stroke. They can be all sorts of things like the size of the hoop. But the point is that from our perspective, the number of transformations that you would apply is much less than the space that the digit's living in, right? So that's the manifold hypothesis. And the idea is that this high dimensional data that seems very problematic just lives on this low dimensional manifold. Now, where is that hypothesis being shown to be true? Like, I've been working on these things for about 20 years and always felt this makes a lot of sense. But, with generative AI techniques, people are really showing this in some sense that, natural images live on these things that are generated by these variational autoencoders or diffusion models or whatever else. And each one of them is reducing the dimensionality of this potentially very, very high dimensional space down to something lower dimension. So, the space, and what you're doing when you use language models is you're triggering it to enter the right region of the space and generate from there. So what's interesting is this departs strongly from this theory, which is really an irritating theory, which you'll read in many, many textbooks. I think it's an irritating theory because I think it's got some nice math behind it. I mean, it's all correct, but there's this thing called the curse of dimensionality. Who's heard of that? Okay, so the curse of dimensionality says that in high dimensions, what we'll get is a situation where our data is all equidistant from one another. So in low dimensions, if I sample from a Gaussian distribution, and this is kind of hard to believe because it feels like, I get some samples like that, right? Oh, sorry, yeah. Thank you. So I get some samples that are all near to each other from the Gaussian. But as I go higher and higher dimensional, what they say is that at one standard deviation away from the mean, all the data will start to sit on that shell. Now, do you find that hard to imagine? So there's basically, when you go very, very high dimensions, you find no data near the mean of the Gaussian. Do you find that hard to imagine?
in a very, so a Gaussian distribution, which let's remember, looks like this. There's the mean. The most probable place in the Gaussian is by the mean. But in high dimensions, there's no data there. What's going on? It all turns out it moves into a shell that's one standard deviation from the mean. I know, it's confusing, isn't it? Okay, but so one of the things about a Gaussian is this could be, Gaussians have this weird property that if we project down any dimensionality of Gaussian down to lower dimensions, what you'll see is still Gaussian. So if I take a three-dimensional Gaussian and I slice through it and project everything onto that slice, what you will see is going to be a two-dimensional Gaussian. But let's reverse that. That means that this implicitly is a two-dimensional Gaussian. It's also a three-dimensional Gaussian. If it were a three-dimensional Gaussian, we would be looking at projection of some data coming from this third dimension being projected down onto that mean. Yeah? Make sense? Oh, well, no, it's a four-dimensional Gaussian. Oh, so that's happened twice now. No, it's a five-dimensional. Oh, that's happened too. Okay, whatever, it was a 25 million dimensional Gaussian. Right? You see where I'm going with that? You're seeing data on there because it's being projected from a distribution from another dimension down onto that dimension. But really, it's not there. It's not at the mean. It's away from the mean. And what it actually does is it ends up sitting. Of course, it's in some sense, it's nowhere because the probability of being anywhere is. But what happens is you can do some analysis, and this also happens for non-Gaussian things. For any setup where you've got independent data dimensions, so you're looking at some data set where you've got, I don't know, dimension 1, 2, 3, and you sample independently from some distribution, as long as central limit theorem applies to that distribution, you will end up with this effect. And the effect has multiple sort of things going on. What it means is that it's sort of like everything is down an axis. So conceptually, the way to think about it is, I can't draw it properly, but conceptually the way to think about it is the data starts flowing around like this and all becomes equidistant from each other. And you can see that in this plot, because in this plot, what I'm doing is I'm sampling from a high dimensional Gaussian, a 1000 dimensional Gaussian, and I'm measuring the distance between 2 points in that. And I'm showing you the histogram of those distances. And what you can prove is that as you go, dimensionality goes to infinity, not only does the points move away from the mean, all the points are equidistant from one another. So the points end up not close to anything and not far from anything. And everyone's, oh, curse of dimensionality, curse of dimensionality. Rubbish. This doesn't happen because data sets aren't formed of data where the features are independent. Of course, it does happen. And what it happens to is what we typically call noise. So noise is of this form. So noise is, the assumption about noise is, oh, well, it's independent, right? So if I think about my output is y equals f of x plus some noise, where sigma is drawn from a Gaussian with zero mean and independent variance. Oh my goodness, that's the thing I've been drawing. That's specifically that thing. So what most machine learning algorithms, I think, is it, what's the guy who does 538 in America? Is it Nate something or other? He calls it, oh, separating the signal and the noise. Well, this is exactly what that thing is trying to do. Is it's trying to sort of separate out the bit of the thing that is subject to this high independence where all these distances are a long way apart. But if we look at data sets in practice, so here, what I've done is I've taken a latent variable, I've multiplied it by a matrix, and then I've looked at the distances there. And what you see is the theory is off, right? So the theory doesn't match anymore. Somehow my theory line's not working, but the theory should be really peaky here, just like it was here. And the theory's off because the data is implicitly low dimensional. Because if the data is sitting on these lower manifolds, you don't see these distance things because nearby points are nearby, right? Because they're living in a 6-dimensional space or an 8-dimensional space, not a 1,000-dimensional space.
I mean, I think people go on about it. I mean, it is an important effect, but it's just not as effective as you might think. So let's see, that was a different point there. So I thought I had that for the oil data. So when you look at real data sets, something fun you can do, and actually I've never seen people do this so much. You can also look at the gram matrix, which is an equivalent thing. But if you were to histogram, and maybe the reason people don't do it because there's a lot of calculation to get this histogram for a large data set, is if you look at the histogram of the square distance between points, if it is really peaky, yeah, then there's no structure in the data. Someone's given you noise, right? But they won't typically have given you noise, and there will be some points, even in very high dimensions, which are very close to each other. And that's indicative of these manifold structures or clustering structures that are going on behind the data. So the sort of notion of, yes, cursive dimensionality is a real thing, but the way people go on about it is a little bit exaggerated, in my opinion, because they talk about it as this, this means we can't model high-dimensional data. Yes, we can. We can model high-dimensional data. If it's got no structure in it, we can't model it, right? That makes sense. It's just got lots of entropy. There's no signal there. But when there's structure there, what we're looking to do with dimensionality reduction is to seek out that structure with a set of different methods. So the sort of other method that we'll mention briefly and continue with next week is, sorry, pull up the slides here, is principal component analyses. And that's how I've shown these. So I've taken this 3,000 dimensional data and I've projected it down onto the first two principal components of the data. And principal components have an enormous number of different sort of motivations. I think they must be the most motivated algorithm in the world. They've got many interpretations as to what the principal components represent. One of the most commonly taught, have you been taught principal components analysis? No? Yeah? Oh dear. It should be taught like first year. It should be the first thing you learn about if you're going to touch data. You should not be allowed to touch data if you don't know PCA, would be my thing. In fact, to my mind, PCA is the computer scientist equivalent of switching it off and on again. Data scientist equivalent. You know, any one time your mum comes to you because you're a computer scientist, or your aunt or your dad or your grandfather saying, can you please fix my computer? The first thing you say is, have you switched it off and on again? Have you shut down the application and restarted it, right? Clears the memory, make sure that it's not something silly going on, right? Anytime anyone comes to you with any data set, your first thing should be, have you done PCA on it yet? Can you show me the first two principal components? It's not, by the way, and this is another enormous misunderstanding around visualization. It's not that PCA is a good model. As we'll see, PCA is actually a very poor model of this data set because PCA assumes that the data is Gaussian in this space and is a sort of Gaussian mapping to a linear space. So it's like a projection where you go from a low dimensional to high dimensional with a Gaussian mapping. It's just that you can see the structure of it. You can see that it's circular. It's a way of getting it down to 2 dimensions and visualizing what's going on. So like I said, before I visualized this data set, I didn't know it was a circle. As soon as I think, oh yeah, that's right, I guess that would be a circle. And PCA is interesting because we'll teach a latent variable interpretation of PCA, but which I think is one of the best ways to think about it. But it has all sorts of other interpretations, including the one we might argue that we're doing here is just looking for directions of largest variance in the data. So there's sort of the classic interpretation of PCA that you will get taught is sort of that you've got some data set and then you look for direction of maximum variance. So the data is living there. And this is, it's not a very interesting data set because you can already visualize it. And once you've found that direction of maximum variance, you look orthogonally for the next largest direction of maximum variance, and you project your data onto these dimensions. So that's PC1, principal component 1, and that's PC2. And the nice thing about it is it turns out that this is solvable through the eigenvalues of the covariance, so the eigen decomposition of the covariance of this data. So these components turn out to be the eigenvectors of the covariance matrix, and their length turns out to be the eigenvalues of the covariance matrix. You can also think of it as a multivariate Gaussian. So if I draw a Gaussian in one dimension, it looks like that. But in two dimensions, if I draw the contours of the Gaussian, they're circular. They're circular because things are dependent on square distances. If things were dependent on L1 norms, then the contours would look like that, right? Because they're dependent on the L2 square distance, they look like that. If I then lengthen one of these axes, if I scale one direction, I get an ellipse, right? And then if I rotate the data, I get the principal axis of an ellipse. So a multivariate Gaussian is like a hyper-dimensional ellipse type thing. And what you're actually finding when you do PCA is the principal axis of that ellipse. So it's sort of showing you where the data is sitting. Now, the data would not look anything like this in this space, right? Because this is going to be a 32,000 dimensional thing. So the data itself is going to be some, structure within that space, which we're then visualizing, and it comes out as a circle. Okay, we'll carry on next time with sort of saying what the sort of underpinning model of that is. But the basic lesson is, from this piece so far, is do either hierarchical clustering or PCA or both, right? Typically do PCA. Always do PCA because you can do it for very large data sets, right? Hierarchical clustering gets harder with larger data sets, but it really does allow you to start seeing and understanding the structure in your data. What everyone does, I'm so sick of people coming into my office from various departments coming in saying, well, I've got this data set, so I've applied a variation autoencoder, or I've done a deep neural network. And I say, okay, well, let's try and understand the data. So have you got PCA visualization? Oh, what's PCA? You must never apply a variation autoencoder if you don't know what PCA is. Never. Because PCA is just a linear variation autoencoder. I mean, there's lots of proofs around that. It's another proof for it, that it's a form of variation autoencoder with linear, right? So unless you've done the simple thing first, you must never do the complicated thing. I mean, and this is causing such a waste of time in the field at the moment because so many people are applying very, very complex methods. If you're doing Gen AI, it's different, right? If you're doing Gen AI, you're not going to generate great data with PCA. But if you're trying to do data analysis, you're trying to visualize some sort of latent space or understand some data set, do PCA first, right? Is my main and number one thing. Also look at things like hierarchical clustering. And then we'll come back to how the PCA is working in next lecture, which is on Wednesday. Any questions? So no PCA yet. Don't you do real world data science or something in that course? Yeah, and you didn't do PCA in that course? Oh. Which type of sentiment analysis? Right, naive Bayes. Okay. Yeah. I think the problem is a lot of people don't do a lot of data analysis, and yet and actually that's being taught by people who do language and stuff like that. In language, you would call this latent semantic analysis or something like that. PCA exists in language, it just has a different name. I think they call it latent semantic analysis. And it's got a slightly different variation because it's sensible to, because it's discrete variables. So that may be why. But for continuous data, you really